Hello and welcome to this webinar titled Disposables, Suitability and Process Economy in Biopharmaceutical Manufacturing, hosted by Biopharma Asia Magazine and presented by Dr. Joachim Walter. My name is Sri Patel and I will be the moderator. Now, before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce you to our presenter. Dr. Joachim Walter got his education and PhD at the RWTH Aachen, Germany, and started his industrial career at Boehringer Ingelheim, Germany in 1985. For nearly 18 years, he was responsible for process development downstream processing from laboratory scale to the cGMP manufacture of clinical-grade biopharmaceutical proteins for clinical phases 1 and 2 in a 2,000-liter pilot plant. In 2002, he accepted a management position at GE Healthcare USA as Unit VP Membrane Separation Sciences, and in 2006 moved on to operate Walter Biotech Consultancy. Since he has been deeply involved both in supervising process development, production processes, and in planning and construction of biopharmaceutical facilities in Germany, Malaysia, China, and Turkey. In October 2016, he was admitted to the Foreign 1000 Talents program of the Chinese government. Dr. Walter has published numerous scientific papers and presented at international congresses while participating in the planning and organization of international congresses. He provides consulting and is specialized in biopharmaceutical development and manufacturing, including single-use technologies, drug safety, documentation, and validation. I would now like to welcome our presenter, Dr. Joachim Walter, to join our webinar. Welcome, Dr. Walter.
Thank you, Sri, for the kind introduction. Welcome, everybody, to my presentation. The title is Suitability and Process Economy in Biopharmaceutical Manufacturing for Disposables. Since the introduction of disposables and gaining popularity of single-use technology for biopharmaceutical manufacturing, there is nevertheless an ongoing controversial discussion on the advantages and disadvantages versus conventional stainless steel environment. Here in the content, you will see, or you can see, what the topic for today is about. First of all, the biopharmaceutical business, the challenges with it regarding the design options for the biotech facilities, rationale for application of disposables. Finally, a little bit of view on the disposables and single-use equipment as it is available in downstream processing. And finally, to be ended up with a summary and a conclusion. The basis for a well-founded decision either on the application of single-use technology or stainless steel-based equipment is a clear scope of the designated facility, be it for process development, a pilot plant, or a manufacturing facility. Accordingly, a number of key questions, which notwithstanding sometimes can be difficult to answer, of course, are: what is the production target? What are the probable cost drivers? Is it about saving investment costs or is it saving operational costs? What type of biopharmaceutical shall be produced? Are there designated needs for specific technical solutions? What is the intended scale, quantity of product required? This will trigger an important decision. What is the designated type of production? Shall the drug be produced in a campaign mode, or does the market volume require a continuous production? Would the feasible and available scale of single-use technology allow to manufacture the requested quantity of product? Hence, it is a target, a versatile facility which is designated to allow a fast changeover from one product to the next, or is it a factory of conventional style more appropriate? Before entering the biopharmaceutical business, single-use technology was widely used in other industries already. For example, in the hospital with respect to medical applications requiring comparable quality standards. So the technical premise was given from the beginning, but the general benefits from disposable single-use technologies was to become evident, namely in the conservative industry as in pharma. The driving force for the supply industry was the opportunity to conquer additional sites and new markets for biopharmaceutical operations at cGMP campaign production of biopharmaceuticals for clinical phases. In the biopharma field, it refers to pilot plants where the advantage of single-use technology was explored, followed by those companies and biotech startups which were limited in financial resources and skilled operators. This even includes universities and scientific institutions. The thorough evaluation of those questions is a key to economy. The underutilization of a facility has a negative effect on the overall cost structure and can hardly be compensated. Such risk could be minimized in case of a flexible facility based on single-use technology applications, as fixed costs of a facility are very much related to size and scale. Thus, small facilities have the advantage of reduced fixed costs, for example, investment, operation, maintenance. This advantage, combined with improved flexibility, which should reflect the ease to adopt to new processes or operational requirements, should facilitate a reduced cost approach. And a major contribution to such requested flexibility is provided by some smart applications of disposable and single-use equipment that we'll now come to. Once a concept for the general operation of the facility in question has been established, the selection and choice of equipment has to be made. Their footprint will define the required space, and most likely and preferably they are based on the intended manufacturing processes. How much and to what detail is the intended process defined? Which volumes need to be handled? How many tanks, containers of which volume are required? What scale of equipment to choose? This may illustrate that in spite of process economy and commercial profit, a suitable development and design of a manufacturing process can only be as good as a management forecast for the product's market. After all, a tailored process design is finally decisive for the overall commercial production economy, and this with respect to both the facility investment, size of the facility and scale of equipment, and of course the operational cost of the manufacturing process. That means a degree of automation, consumption of media, buffers, and consumables. The respective construction of a new facility for the manufacturing of a designated biopharmaceutical is not necessarily the typical situation. More typical, the intended process has to deal with an existing plant and shall fit to the existing space and equipment as much as possible. To design and match a manufacturing process is certainly a challenge to process development, a challenge of different degree for different types of facilities. A conventional facility is typically a large facility with a majority of equipment and piping made of stainless steel. The flexibility of such facility may be restricted, thus providing only limited options. That means due to concrete and stainless steel construction, such facility is at best reconfigurable. The processes to be operated are facility driven. That means process development has to consider from the beginning the options and the opportunities of the classic facility. Any deviation from the existing process equipment and or its configuration typically has to be supported by strong arguments, and this may lead to structurally engineered modifications which may even require a time-wise shutdown of the facility. However, to the benefit of a classical facility, there is, technically speaking, hardly any limitation in scale. As a counterpart to the conventional facility, a flexible facility is a rather small facility, with its equipment and piping made of single-use disposable plastic polymers or in form of a hybrid plant configured in a combination of steel and plastic, where the individual parts can be replaced in a reasonable manner, thus providing superior flexibility. The high flexibility provides nearly unlimited options. The plant will comply with variation of scale and scope of different projects and will be responsive to new challenges. Accordingly, the process is function-driven. That means process development can focus on the target product, while the potentially required hardware and equipment can reasonably be acquired and installed. However, such facilities are limited in scale, as there are clear limitations for the respective single-use disposable equipment. The construction of low-budget containments, suitable to house and operate adequately disposables and single-use equipment, opened up biopharmaceutical manufacturing opportunities in areas and countries with limited infrastructure and engineering skills. To date, the history of single-use technology applications is well established, and disposable consumables are available from many suppliers at fairly comparable quality. Their price has dropped due to increasing competition, though one needs to look apart from the mainstream supplier who are keen and still able to keep their high price level. The art of single-use technology is a knowledge and experience-based ability to select disposable consumables according to the application's technical need, but not beyond. A good example may be the use of polymer bags for media and buffer preparation and storage. The market offers, for example, sophisticated multi-layer bags with vapor barriers and single layer without vapor barriers. Both variants have their significance, and it is up to the user to decide on the appropriate application. For example, if the medium or buffer will be consumed within a short time, thus making a vapor barrier obsolete, or if it is designated for storage. The economy of single-use technology often suffers from a lack of detailed process cost analysis. That means missing economic analysis and cost comparison between both technologies. Therefore, again, a number of first questions have to be evaluated and answered. What is the intended process scale? Consider the disproportionate increase in cost of disposables with scale, namely calculating if there are real savings in initial investment. How many batches? Perform a proper cost calculation on potential cost savings in reduced cleaning validation, and if there is a real saving on cleaning validation using conventional equipment. Continuous production or campaign production. Is the turnover time in between change of product really time critical? Missing economic analysis and cost comparison between both technologies. What are the real savings in initial investment? What are the cost savings in reduced cleaning validation? What are the costs related to testing of disposables for product compatibility? Polymers are suspicious regarding leachables and extractables. Despite intense and thorough testing by the supplier, the individual process solutions need to be tested accordingly, raising the questions as what are the costs related to testing of disposables for product compatibility. A significant economic risk is underestimating the cost of long-term dependency on consumables once the decision has been made to solely rely on disposables and single-use technology. The selection of single-use technology shall be performed in an extremely thorough manner. Neglecting the dependence on single supplier once a single-use equipment has been purchased definitely can be fatal. So now we come to the disposables and single-use equipment to be used in downstream processing. The last but not least aspect with respect to a responsible application of disposables and single-use technology is the management of the resulting tremendous amount of waste. That is what we have to deal with. So the typical downstream unit operations include normal flow filtration, depth filtration, sterile filtration, virus filtration for viral clearance, all that is used since long time. Tangential flow filtration, maybe within scale limits. Centrifugation, maybe not the first choice, for disposables. Chromatography and absorptive separations become more and more possible. But back to the environmental impact of single-use and the waste management. This daunting mess justifies a fundamental concern for the wide use of disposables and single-use equipment with respect to the potential impact on the environment. While with conventional stainless steel hardware the environmental impact is due to the large volume of aggressive fluids for cleaning, the major impact by single-use technology is solid waste of polymer consumables. Other solid waste, such as packaging, paper, cartonnage, and disposable and single-use items like garments, gloves, and so forth, remain the same for both technologies. However, with single-use technology, the saving from fluid waste is only related to cleaning agents. The other process fluids, including sodium hydroxide, for example, for sanitization of manually assembled tubing, membranes, filters, and chromatography columns, resins, remain the same as they are an integral part of the downstream processing per se. The benefit of reduced cleaning fluid and saving from cleaning validation may even further be cannibalized when using hybrid equipment, for example, in chromatography and tangential flow filtration operations. These circumstances should seriously be considered once a cleaning validation argument is used against conventional stainless steel equipment in favor of single-use technology. Accordingly, the environmental impact of conventional technology and single-use technology is largely comparable. The benefits of single-use technology regarding cleaning fluids are minor regarding environmental impact and only major regarding respective single-use technologies marketing by the suppliers. The significant energy consumption in the manufacture of disposables is hardly argued, though this serious negative balance draws a dark shade on single-use technology and may put single-use technology inferior to environmental protection compared to conventional technology. After all, the environmental burden is just translocated from the customer to the single-use supplier. The use of polymer bags for media and buffer preparation and storage is with no doubt the most widely spread application of disposables in biopharmaceutical processes, and it is probably the best example to demonstrate the advantages of single-use technologies. We can achieve an optimized buffer makeup schedule. Buffer preparation becomes feasible at times of low process activities. Buffer makeup ahead of time for several batches. Buffer aliquots of appropriate volume for designated unit operations, and beneficial logistics during the production process itself. And it will help to reduce buffer makeup expenses. Buffer makeup at bulk quantity and subsequent aliquotation. Buffer makeup is reduced to a single operation. It's a one-time weighing of buffer ingredients, one-time documentation, one-time in-process analytics. And it may help to improve process safety and robustness. Filter capsules, bags, sterile connectors, and filter transfer steps are available for safe handling. A most discussed deficit of single-use technology is process chromatography. But first, let's have a look to what is available here from the bags. There are liners which can be used with stainless steel tanks, two-dimensional bags, or three-dimensional bags. It looks nice, it looks neat. However, we have to be aware that the containers definitely are a question. The good thing is that with regarding to storage containers, there are little validation bags typically available where material compatibility with process fluids and product can easily be measured. However, creating space, we have to look for storage space, because finally all these bags, all these containers easily can end up in a mess if there is not appropriate storage space available. The most discussed deficit in single-use technology is a single-use approach to process chromatography. Technically, the chromatographic hardware, that means piping, tubing, inline sensors, columns, are not considered in general as critical components per se for single-use applications. Economically, however, the asking price for those is yet another question. Ostensibly, the price per liter of matrix seems to be the prominent cost factor for chromatography and is assumed to be the typical cost driver of a chromatographic unit operation. For example, affinity chromatography using protein A matrices for the purification of antibodies. But is this really true? The answer to this is quite ambivalent. It depends. Firsthand, it seems to be the price for the investment into a chromatographic resin for the purpose of single use only. Numerous young and innovative companies in Europe as well as in Asia have developed new and/or alternative products which can help to significantly reduce the cost of goods, thus providing obviously an interesting basis for single-use applications. Despite, in case of chromatographic applications, the term single-use may be altered somewhat. A fair pragmatic approach is a single use of resin for an entire fermentation batch in such way that the chromatographic operation is cantoned to typically 3 to 5 cycles, depending on the feasible time schedule of the facility. This will not only significantly save chromatographic resin, but also allows the use of much smaller process equipment, for example, column and chromatography system, with the benefit of reduced footprint and additionally saving investment costs in hardware. After all, taking into account reasonable scales and suitable manufacturing targets, the doors have been opened widely due to the supply of appropriate low-cost matrices and feasible hardware. One example is shown here. It's a system built in China, very nicely made with high-quality sensors, equipment from Europe, from the US. It's providing a single pass in stainless steel, as well as the opportunity to add on disposable flow pass, using the identical sensors, using the identical equipment, the hardware. So that means dropping the price drastically, because there's no need to get a system either or, but to have a system, a convertible system, which allows both conventional and disposable flow pass. Definitely this is the way to go, especially in pilot facilities where there's always a question what to do. Shall I go conventional? Shall I go disposable? So solutions like that are available on the market, one just has to look for. Similar considerations apply to pricing of high-priced resins in general, for example, expensive mixed-mode media or high-resolving uniform beads. The price concern for most other chromatography resins, like ion exchange and hydrophobic interaction chromatography, is less dramatic than it is, for example, for protein A matrices. But anyway, the purchasing price per liter of matrix is not the sole cost factor for the economy of a chromatographic operation. Additional process-related cost factors have to be investigated in depth and evaluated before or latest during process development. What is the price of matrix per liter? What is the dynamic binding capacity? What are feasible flow rates to achieve desired dynamic binding capacity? How can the dynamic binding capacity be improved without compromising the process economy and performance in its entity, for example, by excess process time? How much time is available for the entire unit operation without posing significant negative impact on the process chain and production schedule? Can the chromatographic operation be subjected to cycle operation? How many cycles are feasible to be performed within the available time frame? Can the entire sequence of cycles be automated? Typical beaded matrices are diffusion limited. That means the carrier fluid and the target molecules have to diffuse into the pores of the resin and out again upon elution, of course. Some chromatographic matrices feature a matrix pore design which allows some convective flow within the bead. Accordingly, their dynamic binding capacity is somewhat less dependent on the applied flow rate. At the example of loading an antibody onto a protein A resin, this graphic illustrates the dependency of the dynamic binding capacity on the available residence time. By extending the residence time, for example, from 2 minutes to 6 minutes, the available dynamic binding capacity increases from about 30 grams per liter to almost 50 grams per liter. Nevertheless, the dynamic binding capacity has to be evaluated in each case, individually for the target molecule and the applicable feasible flow rate. As such, the binding capacities, as advertised by the respective resin manufacturers, always should be treated with care, as the data typically reflect very optimistic conditions. The feasible reality for the own product may be quite different. It is common that different products show different dynamic binding capacities at comparable physical conditions. Accordingly, the screening of different resin from different manufacturers is worth the effort, particularly with regard to single-use applications where a reduced resin volume can save substantial investment. The price range of protein A affinity resins, as shown in this table, covers advanced matrices which are supposed to be within narrow limits sodium hydroxide tolerance. We can see, depending on the number of cycles, here shown in blue, in pink, and in green, 3 cycles, 4 cycles, 5 cycles, all 3 requiring the same column, a diameter of about a 60 centimeter column, end up in a quite different volume requirement, 80 liters versus 60 liters versus 50 liters. And if we consider the price range for a comparable protein A matrix from about $6,000 to sometimes up to $18,000. It's a tremendous investment which needs to make, which ranges from $500,000 to $1.5 million or $300,000 to almost $1 million. At, let's say, the typical large target of about 30 grams per liter or 30 milligrams per milliliter. How can we come clear here with disposable or single use of chromatography? A revolution was the last year's change of the FDA's policy, now very much supporting their approach and the support by the US FDA for application of continuous processing in biopharma, which helped dramatically to enforce this in the pharma industry already. Continuous processing is nothing new. Many other industries do this for years, for decades already, but for the biopharma, it's something which is maybe considered quite new. But it definitely has some tremendous advantages when it comes to chromatography. It has specifically become interesting for process chromatography, now often referred to as continuous chromatography, which unfortunately, or of course, is a wrong and misleading term. It is continuous processing using designated chromatography systems, while the individual chromatographic separations follows the traditional principle of equilibration, load, wash, elution, and regeneration, and this in fact is not continuous. Despite the nomenclature, the principle of using sequential columns is quite attractive. Any product which passes unbound a first column is recovered by the subsequent column. Accordingly, the breakthrough of product is no longer a serious concern, and while it allows to apply high flow rates, it additionally increases the dynamic binding capacity significantly. So we better call it the coordinated column chromatography because it is not continuous. It's continuous processing using coordinated column chromatography. A significant portion of the maximum capacity shown here in dark blue on the left part of that drawing of a matrix remains unused, even at a breakthrough of 10%, here shown to the right. Nobody really wants to accept a 10% breakthrough, so typically the breakthrough, what is accepted is in the range of about 5%. So with that regard, the operational capacity of the matrix is definitely very low compared to what one has paid for. So regarding multi-column chromatography, that means using 3 or much better 4-column, the used portion of the matrix increases drastically. Typically, it is more than doubled. And if we compare this now to our previous comparison, in the typical way, ranging from 3 to 5 cycles in a conventional environment, we can see the dramatic impact of the multi-column chromatography, here shown for 3 columns, where we can already reduce the amount of volume for the protein A down to almost 12 liter. And if we do this for a consideration for a 4-column chromatography, then it is about 11 liters compared to 80 liters, 60 liters, or 50 liters running in 10 cycles, while the load target can be increased and improved up to 70 grams per liter. Nevertheless, the matrix has to be cleaned in between the cycles. And although expensive protein A matrices are advertised as sodium hydroxide tolerant, at least for 15 minutes exposure at 0.1 molar sodium hydroxide, these photos clearly show that an effective regeneration will require almost 1 to 3 hours when one is treating the matrix with 0.1 molar sodium hydroxide. Very obviously, sodium hydroxide at lower molarities is not necessarily the perfect solution. In case of low volume, sequential columns operated at higher cycle number, the development of alternative but effective regeneration procedures is worthwhile and feasible. So here in this case, we can see that the so-called sodium hydroxide tolerant protein A still drop in their dynamic binding capacity with regard to the, let's say, old-fashioned protein A, which has a clear drop compared to those. So definitely there is an advantage of the matrices, but the question is, what use can be made of this advantage? So if we have now looked to a so-called sodium hydroxide sensitive protein A resin, an old-fashioned resin, and we are thinking of single-use chromatography, here shown for a 2500 liter scale with about 7.5 kilograms of antibody, we can see that even beyond 20 cycles, we have a very, very good stability, despite the fact that this matrix is not sodium hydroxide tolerant. And if we are increasing the cycle number of the coordinated column chromatography using 4 columns up to 20 cycles, the cost of that matrix is down to something like $20,000 only, compared to the $400,000 to more than $1 million in the conventional processing. So that means the former cost driver protein A can become less expensive than other downstream processing unit operations, like virus filtration, for example. And then it becomes quite feasible. So to conclude and summarize this, disposable consumables are available from many suppliers. Therefore, price competition can be in favor of the user. However, due to lacking standards of intercompatibility of single-use consumables, is rarely given as a commercial key property of single-use technology, often is its design. Such equipment is specific to individual suppliers and their designated equipment design. This enforces a common marketing feature, namely to provide the reusable equipment at very reasonable prices, while the real, that means a profitable business, is in the follow-up business of single-use but expensive consumables. Lashed with the respective hardware, the user has pretty limited options then when negotiating the purchase of the required consumables. So one definitely has to be very careful what to use, but if one is using it the right way, it definitely can be very much of advantage and of economical benefit. With that regard, I'd like to thank you for your attention. And if there are any questions, you always can reach me also at my address. Just drop in an email or so. Thank you very much.
Thank you very much, Dr. Walter. So at this point, I would like to begin the question and answer session. Just as a reminder to our audience, if you have any questions for Dr. Walter, then please send them through to us via the questions tab at the bottom of your screen, and we'll ask Dr. Walter to the questions. So the first question we have is, what kind of bags can you recommend for media and buffer handling?
Well, as I already had pointed out, there are various bags on the market, and they are differentiating very much in price. But the difference in price does not necessarily reflect that the ones are, let's say, not suitable, the sheets are not suitable or whatever. No, it's just the case if I have to prepare a buffer and I'm going to use the buffer within the next couple of hours, vapor pressure, through the bags is no longer of concern. So that means, for example, simple polypropylene bags, low in cost, can do a great deal with that respect, while if I have to prepare buffers or specifically samples, for example, small volumes for analytics, then of course has to be selected. That means I just have to think about what is the purpose of my doing, what is my goal, what do I need, and I may not want to spend money on something which is totally over-sophisticated for what I'm going to do.
Thank you very much. Our next question, is the quality of low-cost protein A matrices suitable for cGMP production and biopharmaceuticals?
This is a clear yes. So I have had the personal opportunity to investigate a number, probably everything was in the market with a protein A resin. And the interesting point is, just look, let's say, to the prices of biopharmaceutical proteins. So we are talking about of high qualified antibodies nowadays to be produced at $50, $60 per gram, and that in a pharmaceutical quality for these products. And protein A is a very, very simple protein produced in bacterial cell culture. So the cost of a gram of protein A is almost in the range of $10, $15 at most. So based on this production cost versus the matrix cost, we can assume that the production cost of a liter of protein A matrix in a commercial environment may be in the range maybe of $100 to $150 at most, Well, then of course there is a packaging, there is a quality control, there is marketing and so forth. Let's say the cost of that kind of product is in the range of $200, $250, and then seeing that even the so-called cheap versions of proteins are being sold at prices of $4,000, $5,000 per liter, and the very sophisticated ones being sold beyond $10,000, you can see what profit there is in, and even there is a tremendous profit with those producing, so to speak, the low-cost protein A. And with no question, it's pretty easy to keep the high-quality standards even of the very expensive protein A, because there is such a wide range of profit, which allows definitely to do that absolutely state-of-the-art and still being able to sell it at a much lower price while making a tremendous financial profit.
Thank you very much. Just another quick reminder, if you've got any questions for Dr. Walter, then send them to us via the questions tab at the bottom of your screen. So, the next question we have is how to avoid the very expensive sterile tubing connectors. What are suitable alternatives for connectors in disposable DSP?
Yeah, that's definitely a good question, because that's a question which typically comes up quite often. Again, the problem is if one gets, let's say, prepared tubing bags supplied, equipped with tubing, typically the individual supplier, of course, equips that kind of equipment, that single-use equipment, with their own connectors. So if I have chosen one supplier, I'm more or less bound to use also products from the other products from that supplier. These connectors are typically quite expensive, especially when we are talking about sterile connectors, which can be used in a non-sterile environment. However, a very simple solution are these sealers, the tube sealers. They can also be operated in a non-sterile environment. They are available from various manufacturers, and then making these even sterile connections in a non-sterile environment by using tube sealers, tube fusers, is definitely a very, very interesting and low-cost way to overcome the very expensive tube connectors. Becomes more and more popular.
Thank you very much, Dr. Walter. So it seems like all the questions that we have today. I would like to take this opportunity to thank our presenter, Dr. Joachim Walter, for sharing his knowledge with us. So audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone.
Thank you. Bye-bye.